osmotic solutions and types of osmosis. So we all know what is osmosis. Osmosis is something where two aqueous solutions are separated by semi-permeable or selectively permeable membrane. Where in the semi-permeable membrane, the solutions will be transporting from its high water concentration to its low water concentration, and through it only water molecules will move. Water molecule will move to where? To its high concentration to low concentration across the selectively permeable membrane. It is impermeable for solute molecules that we need to understand. So if we think about this is a jar, so across the jar, if this is a selectively permeable membrane, so molecules will move from its high water concentration to its low water concentration. Here H2 amount or water amount is high, here H2 or water amount is low. So that means this is a dilute solution and this is a concentrated solution. Now coming to the next thing. Let's see, when a plant or animal cell is placed in a salt or sugar solution in water, the movement of water molecules across the plasma membrane will depend upon the concentration of the solute surrounding solution. Based on this, the solutions can be defined three types: one, isotonic, hypotonic, or hypertonic. So that means the scenario can be occurred if this is a jar and over here we place the cell. So something happens that here the solute concentration is high and here the solute concentration is less. So that means here the water molecule is less and here the water molecules can be more. So things can happen that water comes out. So depending upon this type of scenario, this can happens vice versa as well. So depending upon this, the solutions can be divided into three types: isotonic, hypotonic, and hypertonic. Coming to next, if we can see that this is something, so this area having this thing. So if this is an isotonic solution, so that means here solute concentration and here the solute concentration are same. So that means here the water molecule concentration and here the water molecule concentration is more of same. So it's an isotonic scenario. So that means everything is normal here. But if we can see over in this scenario, what is happening? Here the solute concentration is less and here the solute concentration is high. So that means here water molecule is high and here the water molecule is less. So this is some scenario. What happens in this case? In this case, the water molecule comes from external region to the internal region. So that is called a hypotonic scenario. Hypotonic scenario, what happens? The cell will be turgid. Why? Because from the external region, water is coming inside of the cell. So the cell will be swollen. It gets turgid. But in hypertonic scenario, what happens in the outer environment? Solute concentration will be more. In outer scenario, here the solute concentration will be more and the water concentration will be less. Yes, the cell, what happens? The water concentration will be more and solute concentration will be less. So in type of scenario, what happens? The water will go out. And what happens when the waters are going out? The cell will shrink. It will show a shrinkage. In this scenario, animal cell can burst. But the plant cell will never burst because the plant cell having cell wall. It will be having cell wall. So this scenario can be happens in the hypertonic case. Yes, shrinkage can take place. If we are talking about isotonic medium, what happens? Everything is getting balanced. Same concentration is getting maintained. 0.9% of salt solution is isotonic. So over here, what is happening? See that the water molecules will pass across the plasma membrane in both the direction in equal number. So there will be no change of the cell. Coming to next, what is the hypotonic solution? As we all know, here two osmotic solutions has been given. One having less solute, more water, it's called dilute solutions, and another the cell is placed to a hypotonic solution. Hypotonic means what? Higher concentration of water will take place. Hypotonic. Break the word. Tonic means tonicity. Tonicity means solute concentration. Solute concentration. Hypo means less. Less solute concentration. The cell has been placed to a less solute concentration medium. So that means over here water will be what? Water will be more. So that means when we place the cell in this type of scenario, what happens? Water will comes inside of the cell. Water will comes inside of the cell and cell will gets turgid. It will get swollen because all the water coming in, and that process is called as endoosmosis. What happens? We put a cell to a medium where in this medium what happens? The water molecule is high and solute molecule is less, so that this medium is called hypotonic because tonicity is depend upon the solute concentration. Here what happens? The cell will get turgid, all the water coming in and the cell will get turgid and it will result to endoosmosis. So that is called hypotonic solution. In the scenario you are seeing what happens, the cell will gains water by endoosmosis and it gets swollen. Coming to hypertonic solutions, according to the name we all are understanding, hyper means more, tonic means solute concentration. Solute concentration is more. So that means what happens? In two osmotic solutions, the solute concentration is high. That means it's a concentrated solution, and here water concentration is very low. That means it is 1.25% solute concentration or 10% of glucose solution. Right? If a cell is placed over here, what happens? The cell will be having water high and solute less. It is placed to somewhere where solute is high. That means it's a hypertonic and water is less. So what happens? The water will come outside. Water will come outside. So what happens? When the cell lose water, it will shrink and reduce its volume. The water molecules leave the cell. What happens? It will shrink and reduce the volume. It's called exosmosis. So endoosmosis will be happening in the hypotonic situation where water comes in the cell. Hypertonic situation, what happens? Water goes out from the cell, and that's called exosmosis. You can see the cell loses water and it will get shrinks. Now we have to study the endoosmosis and exosmosis of a resin. So 4 to 5 dried resins we put in the water. So you can see that there is dried resin we put. Now what happens when we put it to a sugar concentrated or salt concentrated solution? What the things will be happening? Think about if we put it in a salt or sugar concentrated area. So what happens? The exosmosis will take place. So think about this is a resin. We put it to a salt concentrated solution. To a salt concentrated solution. What happens? Over here, this area will be solute high and water less. So water will comes out from this resins. What happens? The resin will shrink. The resin will be shrink. Exosmosis will be taking place when we put it in a concentrated sugar or concentrated salt area. But if we put the resin to a normal water condition, what happens? The water will come inside the resin, and what is happening? Endoosmosis will be happening and resin will be swollen. What happened? Resin will be swollen. So the resins will be swell up when placed in water due to an solution, that is exosmosis. So this is the whole scenario. We understand about hypotonic, hypertonic and isotonic media. That is the different types of osmosis.